Okay, does anyone have any questions regarding lipids or transport that we covered last week? Oh, no, we covered hemoglobin and myoglobin as well. We finished with transport. Any questions on those two last topics? Any questions? So there's a quiz that's due tonight at 10 o'clock that started last Wednesday. There's no Dropbox assignments due until after the exam on carbohydrates, which we're just starting carbohydrates today, so there's nothing due right now except for the quiz. Oh, achieve is due. Yeah. Yeah, the 7th, achieve. I just want to make sure I call it the right program. It's other biochemistry. And then it used to be called something else. So it's, yeah, so that's due the 7th. We haven't tried it yet. Okay. All right, so we're starting carbohydrates today. And then in a week from today is exam 2. And the study guide is under the study guide module for exam 2. So be sure to let me know if you have questions. You can also email me if you can't come to office hours or if you'd rather not ask in class. Yes.
For this exam, are you also going to be on Zoom again?
Yes. Yeah, so that's right. Thanks for reminding me. So if you have questions during the exam time, or which is class time, then you can either visit me on Zoom or email me. I'll have email open and Zoom open at the same time. I usually have 3 devices in front of me when I'm off. Yeah, so I'll be there for your questions. And then I can always share the screen with you. And then always, have your calculator and scratch paper there in case you need it. Not that you're doing a graph, but you have to analyze a graph. Might be good to have a ruler to put up to the screen to determine Vmax or something. It's more of a choice, but still. Other questions? Not at the moment. Okay, study guides available. Okay, so I'm recording this as well. Although if you go to gallery, you see the ones from the old semester, previous semester as well. Seems like there's no airflow in here, right? Okay, so we'll start carbohydrates. It's working. And your book always has to show you something. So they're showing a field of wheat because that wheat products are carbohydrates or polysaccharides. So sugars have this molecular formula. They're basically hydrated carbon. Okay, so they have a lot of hydroxyl groups, we'll see. Because they have hydroxyl groups, they can hydrogen bond with water. And so we're going to see eventually, once we get through aerobic respiration, which usually comes after the carbohydrate chapter, and once we cover beta oxidation of fatty acids, you'll actually see that since carbohydrates are more oxidized than fatty acids, that we do get more energy per carbon per fat versus per carbon per carbohydrates. But sadly, your body's not really made to break down fats on a daily basis like carbohydrates. So carbohydrates are involved in energy, which we will see during the cell respiration or aerobic respiration exercises or notes, as well as structure. They make up cell walls. They can be involved in cell-cell recognition. Okay. You may recall that the plasma membrane is not only made up of lipids and proteins for transport, but there are sugars usually on the outer surface of the cell, and that helps white blood cells distinguish self from non-self, and then that can lead to the immune response. And then white blood cells are like, okay, that's a bacteria, not this person. And we're going to see that sugars exist in 2 forms: an aldose and a ketose. Aldoses are aldehyde sugars, ketoses are ketone sugars. Okay, and we'll discuss the 3 carbon ones first. We'll also then cover mostly 6 carbon ones. So I focus only on, for the monosaccharides, I focus on the 3 and 6 carbon sugars because we'll see those in glycolysis in the next 7 lectures. But I will point out the other sugars, but we focus mostly on the 3 carbon and 6 carbon sugars. So this is just a reminder of functional groups, although I think the first week I could tell you knew your functional groups, but it's good to review. So a carbonyl group is just the carbon with the double bond oxygen. An aldehyde will have 1 hydrogen and then another carbon on the other side. Ketone has 2 carbons on either side of the carbonyl. Now carboxylic acid isn't shown here, but you probably remember that's similar where this would have an oxygen here. Okay, so these are the simplest sugars. They have to be at least 3 carbons big. And we will actually see glyceraldehyde and dihydroxyacetone, but in phosphorylated forms in glycolysis in stage 2 of glycolysis. So we won't begin glycolysis. Yeah, that's not scheduled until after next week. So we have dihydroxyacetone and glyceraldehyde. This might remind you of glycerol, which is one of the building blocks for glycerol phospholipids and triglycerides. Okay, so you might recall that glycerol has carbon and a hydrogen and a hydroxy group, and then this is CHOH, and then carbon number 3 is CH2OH. But for dihydroxyacetone, carbon number 2 is oxidized to a ketone. And then glyceraldehyde looks like glycerol, except for carbon number 1's been oxidized to an aldehyde. So if you can make those relationships, then it's not like learning something new all over again. Now, since dihydroxyacetone has no chiral carbons, there's no mirror image of it. Okay. Whereas diglyceraldehyde, there is a mirror image because this carbon is chiral. So they're like facing a mirror here. We did talk about D and L sugars, not sugars, amino acids a while back. So we talked about D and L in the previously. But here, and we'll see that with these 6 carbon sugars or anything with chiral groups, you can have D and L sugars. Now for amino acids, the predominant form was the L amino acids, but for sugars, the predominant form is the D form. And those are the only forms we're going to learn. And we're going to learn some terms for the isomers. Okay. And so we'll be looking at some pictures. So as you may have seen with glyceraldehyde on the previous figure, there was at least 2 carbons that had a hydroxyl group. Okay. And then the middle carbon was a ketone. And we're going to see when we study the 6 carbon sugars, there's multiple chiral centers. So there's multiple opportunities for each of those carbons that's chiral to be in the D or L form, or have the hydroxyl group on the left or the right. Okay. But when we're determining D or L, we're going to see that even though it may have at least 5 chiral centers, the chiral center that you look at for D or L is the one furthest away from the carbonyl, okay, which we will see. I get ahead of myself. So when we showed you the glyceraldehyde, the D and L, where the right, the L form had the hydroxyl group on the right, and the D form had the hydroxyl, and the D form had the hydroxyl on the right, L-glyceride had the OH on the left. Those are known as enantiomers. So mirror images are not superimposable on themselves, okay? And they have a term for those when they D and L called enantiomers. So there's a little bit of vocabulary here. This is looking at various sugars here. So they're talking about glyceraldehyde and dihydroxyacetone. And sometimes they'll call them constitutional isomers. I don't usually use that term, but if you were to look at the number of carbons, hydrogen, and oxygens, they're exactly the same, but they're rearranged as an aldehyde or a ketone. Okay. Enantiomers, of course, they have the same formula like above, but they're mirror images. Okay, so they're not considered constitutional isomers because they're basically the same molecule. except the hydroxy group has changed to different sides of the Fisher projection in this example. Diastereomers are not mirror images, but they're isomers. Okay. And so we have glucose here, but what's changed here, if we look at it, and they're both D forms. And the reason they're choosing D for these is they're looking at the last chiral carbon, which is carbon number 5, that's furthest away from the carbonyl. Sometimes when you see carbon, hydrogen, oxygen, that's the same as saying carbon with a double bond and a hydrogen. So this is another way that they draw the aldehyde form, usually if they're limited in space. And so these are both aldehydes. So we want to look for the last chiral carbon furthest away from the aldehyde group in this case. If it was fructose, which I don't show a picture of right now, it'd be furthest away from the ketone group, which is usually position 2. So the last chiral carbon is carbon number 5. And if the hydroxy group is on the right, it's D. If it's on the left, which isn't shown here, if it were on the left, it would be L. Okay. And so these aren't exactly mirror images here, but we see that in this case, we have a different orientation for the hydroxyl groups here on position 2, as well as position 3. So there's several that have changed the orientation of the hydroxyl group and the hydrogen, not just one. And so they consider these diastereomers. They have the same composition, but the rotation about these hydrocarbons is different. Now, we'll also look at isomers when you go from a ring, go to a ring form, and we'll learn how to make the ring. And the only difference between alpha and beta D-glucose is what's happening at position 1. Okay, is the carbon hydroxyl pointing up, then it's beta. If it's pointing down, it's alpha, but otherwise everything else is the same. And one of my friends that was a fan of baseball, or still is a fan of baseball, she said to remember for beta, batter up. Means the hydroxyl group will be pointing up. Now my students last semester taught me the other one for alpha. You probably already know it. All fall down. And I said I could relate to that because I'm from here. So because I end up picking up skateboarding or something. You want to see me that often then. So we'll come across that when we learn how to go from the Fisher to the Hayward. Now, some of you may have already had that in organic chemistry, some of you. And then it depends. Some organic chemistry may not have it if they saved it to the end of the semester and then out of time. So we'll be going over that. But we don't learn chair for confirmations in here. I think people are usually relieved when I say that. I wasn't a fan, and then I found out there was stencils later after I finished doing that. But like rulers would help you. But then one of my students says, "Oh, they won't let you use that on exam. They won't let you use the stencil. You're just trying to be neat, but they won't let them use it." So again, enantiomers are another way to say the mirror images, and just reiterating that you can't superimpose them. Epimers are just when you rotate one hydrogen OH pair. Okay, and whoops. So for and we'll see this on another picture, but if we had another D-glucose here that rotated this hydroxyl group with the hydrogen, so they're on opposite sides than they are here, then that would be an epimer, where you only change one carbon. Also, alpha and beta, even though they're called anomers, that's a special case of epimer. But when it's epimer in a ring, they call it an anomer. They have a name for everything. Instead of just calling it epimer, they call it an anomer because it's in the ring. I said that already. Okay. And that goes here, but we'll cover anomers and the alpha and beta when we get to the drawing of it, which won't be yet. And then diastereomers are a special type of isomer where they're not mirror images, so they're not D or L necessarily, but they're just one carbon's hydroxyl and hydrogen have been rotated. So it's an epimer. Okay, I already said that epimer one hydrogen OH has been rotated. And we'll see some examples on the next slide where we compare galactose to glucose, and then we'll also compare glucose to mannose. So they basically have the same number of carbons, which is 6, so they're all 6, C6H12O6. Okay, don't memorize that. And that's not shown here. But yeah, so mannose and glucose are up here, and if you look at them, we see that the aldehyde, well, that's the same. And then carbon number 2, look, they're facing each other. So that's their C2 epimer, okay, whereas everything else is the same, that they're both on the right side. So here, carbon number 2, hydroxyl's on the right versus the left, so they're C2 epimers for glucose and mannose, but they're both these sugars because it's the last hydrocarbon that has the hydroxy group on the right. So what I'm saying, and I will be giving you a list of ones to recognize. Once you learn glucose, you just need it, you don't have to remember them separately like you had the amino acids. Okay. For this one, like once we learn glucose, we'll know that mannose is a C2 epimer. So if you know glucose and then mannose is C2 epimer, you just need to know to rotate the hydrogen and OH on carbon number 2. You know, so you might want to write, you know, if you were going to write it, I do have you do one drawing for the drop box, but it's not here. Because that's not exam 2 2. But, you know, I would just draw in pencil and then you can erase it and switch them. And then we'll learn that galactose is another epimer of glucose. And so once you know glucose, you're going to switch a different pair on that. And then fructose is just a ketone version of glucose, but I'm getting ahead of myself. Okay. We're not going to deal with the 5 carbon sugars here. If we were going to go over DNA and RNA structure, then I would. Okay. But we did talk about glucose and mannose, where mannose again, no, we talked, yeah. So mannose and glucose, yeah, so glucose and mannose are C2 epimers of each other. But galactose, can anyone tell me what epimer galactose is of glucose? C4, yeah, so here the hydroxyl group is on the left, here it is on the right, that's correct, C4 epimer. So once you know glucose, you actually know 3 molecules. Fructose looks just like glucose if we work our way up. Carbon 6 and 5 are the same, well, they're always the same. Carbon 5 hydroxyls on the right, that makes them all D sugars, because that's the one furthest away from the carbonyl group. Carbon number 4 is the same as glucose. Carbon 3 is the same. What happens is carbon 1 and 2 are different just because one's an aldehyde and one's a ketone. So that's where it changes. So now, however, for fructose, you have an extra CH2OH. So it has one less chiral center. Otherwise, it's similar. Just the tricky part is carbon 1 and 2. And those are the ones you're going to be recognizing, but I do have a list for you later, I think, on the notes. But it will be glyceraldehyde, dihydroxyacetone, and then glucose, mannose, galactose, and fructose. So it'd be these 4 plus glyceraldehyde and dihydroxyacetone. Now, if I accidentally say dihydroxyacetone phosphate, I'm already thinking about glycolysis. It has a phosphate on it. But I'll try not to say that too much. Okay, so this goes over how to go from Fisher to Hayward. Okay, and they do use these terms hemiacetal, and then later full ketal and full acetal. I don't really dwell on those names, but your book likes them. So when you add, so if you have an aldehyde and you had an alcohol group to it, and you get rid of that carbonyl group, then you're left with 2 alcohols, and they call that a hemiacetal. And the same with the ketone. If you get rid of the carbonyl and then have 2 hydroxyl groups, then it's called a hemiketal. Some language there. Now, this is kind of strange reasoning, but they use this. So this is going to lead up to drawing the Fisher and Hayward, the Hayward structures from the Fisher. They notice that the 6 carbon Hayward structures or ring structures remind them of a chemical called pyran. So they decided that when you go from the Fisher to the Hayward, they wouldn't include pyran in the name of the ring structures, even though these ring structures of the carbohydrates do not have double bonds. They decided to put pyran as part of the name when you go from a Fisher projection to Hayward structure, or from a linear to ring structure. Similarly, the sugars that make the 5-member ring, they said, oh, it looks like furan. So we're going to, when we call it, when it's in the Hayward structure, we're going to put furan in its name. Now, the only sugar you're learning that will be in the furan, have furan in its name is fructose. So usually 6-carbon aldehyde, 6-carbon ketones will have furan in it. All the 6-carbon aldehydes will be pyran. But it was kind of interesting logic. Oh, since it looks like that, we'll put that in the name. So, wasn't my idea. So, so we will talk about the cyclic forms, okay? And I did mention already that that type of isomer, when you have alpha and beta, that's called an anomer. That's a special type of epimer called an anomer, where instead of it being changing from left to right side, it's changing from top to bottom. And we talked about pointing up as batter up, and pointing down is all fall down. Okay. But, you know, if you want to get more technical, alpha means below the plane of the ring, beta is above the plane of the ring and on the same side as carbon number 6, which has that CH2OH. And this shows how to get to the ring form. So this is glucose. Okay, and so usually to get the big ring, which is the 6-membered ring, carbon number 5, its hydroxyl, particularly its oxygen, is going to attack the carbon. So it's a nucleophilic attack because the carbon's depleted of electrons, but that's as far as I get into mechanism in this class. And and that's shown here. So then it's nice, it's numbered here. So carbon number 5, which was the one that helps us determine D or L usually, that hydroxyl group attacks the carbonyl. And then, so this carbon here, which was the aldehyde or ketone, will no longer be an aldehyde or ketone, but it's very reactive. That carbon is very reactive. And it's also called the anomeric carbon. So like that word anomer, where's that word anomer? Let's see. Oh yeah, where it says anomer, they call it anomeric. They put the IC on it for that carbon, just to remind us that it was the former alpha or beta form, or it's going to be the alpha beta form. So if that hydroxyl group points down, it's alpha. If it points up, it's beta. And then this shows you how you name it now, since it sort of looks like pyran, you know, not exactly. So you say alpha because that hydroxyl group is pointing down. And then we're only learning the D sugars. And it's really hard to determine if it's D or L because now that hydroxyl group is gone. It's carbon number 5 is here. That's carbon number 6. So this is 1, 2, 3, 4, 5, and 6. So it's really hard to tell if that's D or L because it's reactive, because the one you would have used to determine it's D or L is gone. And you can't tell from the others because those are the ones you assigned D or L. So for our purposes, it's always D. And then you say glucopyranose. Instead of saying glucose, you say glucopyranose. So that tells people it's in a ring, even though you can see it's in a ring. But, and then the same as beta because the hydroxyl group is pointing on the same side of the ring as the CH2OH, or batter up. And then this is not on the same side as the ring as CH2OH, or all fall down. And it's just 50% chance which one is going to be up or down. Okay, and that's not the only one that can be in a sugar ring. So we're giving so all the aldehydes would do that, but you're only learning the 6-carbon one. And then we're also going to look at the ketose fructose. So it's carbon that or hydroxyl group that will attack the carbonyl is carbon number 5 as well. But it's attacking carbon number 2, that's where the carbonyl is, so that's why it's a smaller ring. Okay, and that's why it's smaller. But again, that oxygen will attack the carbonyl. This will be, so now we have this as carbon number 1, which is fine. So carbon number 1, this is carbon number 2, that's the former anomeric carbon. And what's going to happen now, they're showing the alpha form with it down. So then, so that's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Okay, now this is showing you only the alpha form, and now how they changed the name is fructofurano's, okay, instead of pyranose, because it's not a 5-membered ring. It's a 5-membered ring. But I put the beta form here, here. So the beta form is a little bit tricky when you number it, because you have to kind of like number it like zigzag. So this is carbon 1 here, carbon 2, so you have to go up and then down. So carbon 1, 2, 3, 4, 5, and 6. So usually if people get confused, it's because this one's pointing down, and they have to remember that's actually still carbon 1, and this is carbon 2, and that's carbon 1, that's carbon 2. Yes. It prefers the ring. That's a good question. Yeah, it does prefer the ring. And then usually when it's in chains, it makes it easier for it to link together. I think that's the reason. Yeah. So that's how their names change. We'll also learn how you name them when there's 2 joined together as disaccharides. Polysaccharides will usually keep their common name, just like glycogen, amylose, and amylopectin. Oh, so I did have a list too. But the only time you're going to, so on the Dropbox assignment, which of course not related to exam 2, so it's, I think I opened them all. So the due dates are not due. So I won't track the thing. So ones you should know, or which means so on exam 3, you want to be drawing them, but you recognize them. But for the Dropbox assignment, I usually have it linked to 2 of these 6 carbons together. So these are the 2 3-carbon molecules you should be able to recognize. And then these are the 6-carbon ones. All of them are aldehydes except fructose, which is a ketone. So those are the only ones. So the only time you're going to draw them is on the Dropbox assignment. And then it will usually be 2 Hayworths linked together when we get there. All right, so now I'm going to explain what reducing and non-reducing sugars are. Okay, but they're using a monosaccharide here as an example. Has anyone done the I don't want to get it mixed up. The Benedict's test? Anyone heard of that? Well, Benedict's test has copper in it, and if you have that solution with Benedict's, it reacts with reducing sugars or monosaccharides and some disaccharides. If that carbonyl group that was a carbonyl group before it became a monosaccharide, it will still react with it, and what happens is the copper in that reagent gets reduced by the sugar, and the sugar actually gets oxidized, so carboxylated. And that's shown here. So this is glucose. I always know it is because it always goes down, up, down. So the glucose, is this in the alpha or beta form? You look at carbon number. Beta. That's right, beta. Is it pointing up? And then they just then they put it back in the Fisher because when you do the Benedict's test, not that we're doing it in here. Apparently there used to be demonstrations in here. So it reacts with copper. Copper gets reduced from +2 to +1, and this aldehyde becomes a carboxylic acid. If it was fructose, the ketone would become carboxylic acid. And so that's where the name comes from when they say reducing sugar, because that monosaccharides, it can be some disaccharides. As long as you have a free former carbonyl carbon as an anomer, free that's not in a glycosidic bond, it can react with properly. And so that's where that term comes from, reducing sugar, because it can react with chocolate and cause it to be reduced. And so all three monosaccharides are reducing. We'll learn in disaccharides how you can distinguish if it's reducing or not reducing. And why do we care about that? Because it's not a lab. They actually based how to name disaccharides based on if you have a reducing or non-reducing sugar in the disaccharide. And then this is just extra information. You may know someone that has diabetes, and you may be wondering what the HB1AC is about, or A1C is about, and that gives a truer picture if someone's diabetic or not than someone they just quickly check their test and the glucose not, because that could just be one day where you ate French fries right before the test. This is more like several, I think a month to 2 months, and then if that's high, it's not just French fries. But interesting enough, we talked about hemoglobin. Well, there's so much sugar around in the bloodstream, and we know that red blood cells circulate the bloodstream. That the hemoglobin in the red blood cell is glycosylated or has a lot of sugar rich on it, and that's an indication of diabetes. If your hemoglobin has been exposed to that much sugar that's attached to your hemoglobin, then that's a sign that you most likely have diabetes. So here, this ester linkage can be found if proteins are attached to the sugars, okay, which we talked about a little bit, but not in great detail. For lipids, some lipids are and proteins can have sugars attached to them, so they can be in a membrane. And if a amino acid is attached to a sugar, it can be as an ester linkage. Okay, and there's only 3 amino acids that could do that, that have a hydroxyl group that could attach to sugar in that way. Does anyone know which sugars, amino acids those would be? Because we know our amino acids to be simple. Anyone know the 3 amino acids with the hydroxyl group? Serine, threonine, one with the big ring. Close. Tyrosine. Very, very good. So, so those will be there. And then what we'll see is in glycolysis, you can have a phosphoester where a phosphate's added to the sugars, like glucose 6-phosphate and fructose 1,6-bisphosphate. Don't memorize this slide, but this is just showing you that they can be modified. I already gave the list of ones we're going to remember or recognize, I should say. So glucose 6-phosphate, again, we'll see in glycolysis. That's the first input of energy into glycolysis. The hydroxyacetone phosphate is made at the end of stage 1 and disappears at the beginning of stage 2 of glycolysis. So we'll see them in the next chapter when we get there. Now we're talking about linking 2 monosaccharides together to give a disaccharide. Okay, and so just like anything we've mentioned before, when we make polymers of amino acids to make proteins, add fatty acids to glycerol, or we're not studying DNA in here, but if you're adding 2 nucleotides together, you lose water. Similar here, if you add 2 or more sugars together, you always lose water when you're adding them together. So they're always dehydration or condensation reaction. Now here, we have carbon number 1 linked to carbon number 4 of 2 identical sugars. And again, they always like to draw glucose. I just know it's down up here. But here we have carbon number 1, which would have had the former aldehyde group, and this is carbon number 4. Okay. And they're just making a big deal about the O-glycosidic bonds. Most sugars are linked together by oxygen from a hydroxyl group bond to the other one. If we're now going back to those terms, reducing and non-reducing, well, all monosaccharides are reducing because that former carbonyl group is not bound to anything else. It's free, it's not interacting with anything. On here, though, if we look at the glucose on the left, carbon number 1, which used to be an aldehyde for glucose, is involved in this bond with carbon number 4 of the second glucose molecule. So this, on the left side, this monosaccharide would be considered non-reducing because it can't react with copper because it's bound to copper, carbon number 4. So this glucose here again would be non-reducing because it can't react with anything because it's bound to carbon number 4. Now, in contrast, this carbon number 1, which is the former aldehyde, it's not bound to another sugar. So it can still react with copper and reduce it. So this would be the reducing end. This would be the non-reducing end. But overall, this disaccharide is considered non-reducing. Okay, so you can have reducing and non-reducing ends. But then the sugar will be considered reducing if at least one of the aldehydes is available, former aldehydes or ketones is available to reduce copper. So you can have, and we'll see that you can have two non-reducing sugars bound together where carbon 1 and carbon 1 would be together for two aldehydes, or carbon 2 and carbon 2 of two ketones, or carbon 1 with a carbon 2 of an aldehyde and a ketone. So both would be non-reducing, but you never have any that are reducing, because there always has to be one reducing carbon involved in the bond. And why am I reiterating that? Because that's going to explain how we name them later. Now, please don't think, don't memorize these as their common names. I'm not going to show you sucrose and you have to identify sucrose. We're going to learn how to name them systematically. Just like the fatty acids, we're not memorizing the common names. We were learning how to name them with the systematic names, like dodecyl. So sucrose we'll learn is made up of glucose and fructose, and that's the strange one where they actually rotated it to do the bonds. But we're going to learn how to draw them vertically instead of horizontally. That's the new thing now, to draw them vertically. I went to school to do horizontally and rotated them if two anomeric carbons were involved in the bond, but we don't rotate them anymore. We stack them, which is not shown here. And we'll learn about lactose, which is glucose and galactose. And maltose is actually two glucoses. But we're going to learn how to name them systematically. These are the common names. We're not going to learn them that way. But some will have a beta linkages because the hydroxyl group of carbon number 1 was up. And some will be considered alpha linkage because the hydroxyl group of carbon number 1 was down. Okay, and then this is going to lead to my next slide. Can you draw sucrose stacked instead? Because there it was the normal horizontal traditional thing. But the new thing is, if we look back at sucrose, and you wouldn't know it otherwise because we're just learning it, is that they have rotated the fructose, so the carbon 2 is near number 1. Normally carbon number 2 is on the other side, but they flipped it. I don't have you ever flip it for that Dropbox item. We just did the new convention, which is horizontal, which is easier, but people think it's harder. I don't know why. You just draw one and you draw the other one and you erase the elements of water and just can. So it's much easier, really. But here it says, can you draw sucrose stacked instead? So think of bunk beds. They're stacked. They take up less space in this. So here, they seem to love glucose a lot. This is a glucose getting down right there. So they took two glucoses, okay, and this one's pointing down. So which one's this, alpha or beta? Alpha, and what's this one? Beta, it's pointing up. It doesn't always have to be that way, but it is a neater vertical structure if alpha is pointing down and beta is pointing up. But you can have them both alpha and both beta, or the reverse. But in this case here, so they're both glucose, and then as you said, this one was the alpha and this one was the beta. And so you just basically draw them, one and the other one, then you can just remove the elements of water and connected. It's not that. What's that?
Is that maltose?
Yeah, that would be maltose, although I don't make you learn it as maltose. Yeah, that would be maltose. Okay, so that's how you do, this was example of two glucoses stacked instead of horizontal. And I think I'll have to check your assignments. I'm having you draw two 6-carbon sugars together, but in a stacked conformation. And then I have you name it, but I haven't gone over naming yet. But I will by the time that's due. Hopefully not today, but it looks like I will today. So we went over what reducing and non-reducing mean. So we're talking about one monosaccharide. We're talking about that carbon number 1, if it's an aldehyde, is free to react with anything because it's not in the bond. If it was a ketone, it would be carbon number 2 that is free and it's not in the bond. Then that one's also reducing. Non-reducing is if carbon number 1, if you're talking about the monosaccharide, is in a bond, or carbon 2, if the ketone is in a bond. But overall, if you have 2 monosaccharides joined together and there is one reducing end, the whole thing is called reducing. If both anomeric carbons are bound to each other, then it's non-reducing overall. But sometimes when you're naming it, you have to figure out which is the reducing end of the disaccharide to know which one to name first and second. So always the non-reducing end is named first, and the reducing end is named second. It's just out, it's like almost like after order. Non-reducing becomes before reducing. And if we go back to this picture, on this one, carbon number 1 is joined to carbon number 1. So this is going to be non-reducing, non-reducing. And we'll see that when I go over to name it. When they're both non-reducing, obviously it's non-reducing because there's no reducing in it. But it doesn't matter which carbon or which sugar you name first, if they're both non-reducing. So if it's a reducing sugar, one end can be reducing. And that would be if carbon number 1 of an aldose is free or carbon 2 of the ketose is free. But if both are aldehydes and both carbon 1s are joined together, like in, let me have an example of those. But if we have 2 aldoses with carbon 1s joined together, then it won't matter, but it'll be all non-reducing. If carbon 2 of a ketone was joined to another carbon 2 of a ketone or ketose, it would both be non-reducing. And if carbon 1 of an aldose was joined to carbon 2 of a ketose, then it'd also be non-reducing, non-reducing. But typically carbons 1 and 4 are joined together when one's non-reducing and one's reducing. So you won't find it in the book, at least on my notes for this, so if you have questions, so first of all, when you have a disaccharide, we're only going to name disaccharides like this. You have to determine which end's the non-reducing end versus the reducing end. So you have to determine that, or you have to determine if both are non-reducing, like sucrose. Also you want to know, so also just like fatty acids, when you name them in a triglyceride, the first fatty acid, or in this case, the first sugar in a disaccharide, we will lose, even though it's going to be pyranose for an aldose if it's in a ring, or furanose if it's a ketone in a ring, that pyranose and ketose suffixes, even though it has pyranose here, if it's the first sugar named in the disaccharide, it will go to pyranosyl or furanosyl. Okay, just like amino acids, when you have a tripeptide, those amino acids lost the INE or YL. Here the same thing. If you have 2 sugars and you're naming one sugar and then the second sugar, the first sugar has a suffix of YL. And then the second sugar will keep its name for the most part. And most of them will end in OSE if it's reducing. If it ends in side, meaning it was non-reducing, then it ends in side. Okay, we'll see some examples. So some disaccharides will end in ose because you still have a reactive carbonyl that's not bound to something. And others, both carbonyls are joined to each other, so the final name is side to indicate both are non-reducing. So we'll look at some examples. So we're not like, I won't show a picture of lactose and expect you to identify it as lactose. Okay. Same with sucrose, but we'll look at how to name these or some of these. Okay, I hope I fixed it. This version's fixed. Okay. So well, let's look at this one first. Maltose first, which someone identified earlier. If we look at this one, the glucose on the left is alpha, and they labeled it here, but on my exams they won't be labeled. It's alpha because hydroxyl group was pointing down. This is alpha over here, and it's carbons 1 and 4 linked together. So which one, the left one or the right one, is reducing?
The left.
So remember, reducing means the former carbonyl is free. So it'll be the right one, actually. So this one's the reducing one, and this is the non-reducing one because carbon number 1's in the bond for the aldehyde. So to name it, that means we're going to name the one on the left first because it's non-reducing. We'll remember that one first, name that one first. Since it was alpha because the hydroxyl is pointing down, you say alpha D, and you want to say glucose, but we can't because it's in a ring, so we have to say pyranosyl. Normally we would stop at pyranose, but because it's linked to another monosaccharide, it's going to be pyranosyl instead of pyranose. And then 1 to 4 means carbon 1 is linked to carbon 4, and they draw an arrow. And then this one is alpha also, it just happens to be, it doesn't have to be, it could have been beta. P, and then it's glucopyranose, and it ends in OSE, which means, hey, the second carbon, or second ring, that anomeric carbon is free. It's not bound to anything. It could reduce copper. So then they put OSE. Now, if we look at this one, lactose, which is galactose and glucose, we see that, again, but usually if one's non-reducing, only one, it's always on the left. Okay. And so carbon number 1 is involved in the bond, so it's non-reducing, whereas this carbon 1 is free to react with copper, so it's not reducing. It's reducing. And so you would name this one first, but since it's galactose, remember we'd say C4 ephemeral. Okay, so if we look at carbon 1, 2, 3, 4, it's up over here, whereas over here it's down. So we have beta, because this was up, beta D, and then galactopyranosyl. And it has sil because it's the first one, then 1, 4, and then alpha down, D, glucopyranose is OSE again, because this second monosaccharide is, this one's free to react with carbon number 1. Now to change the name here. Right, but so we would be drawing this vertically, so we don't have to rotate anything. They had to rotate, but it doesn't look as pretty when it's stacked, but it works. But because both carbon 1 of the aldehyde and carbon 2 of the ketone are involved together, both monosaccharides are non-reducing. There's no free carbonyl group, a free anomeric carbon to react with copper. So they're both non-reducing. You can name it left to right, which I just do out of habit, or you can name it from right to left. It doesn't matter because they're both non-reducing. But if I do left to right, well, this is glucose. It's much bigger than fructose. It's going to be alpha, which it is pointing down. D, glucopyranosyl, just like the other ones. 1 to 2, carbon 1, carbon 2. And we'll have to take the word for it beta because they flipped it. Beta, D, but we changed the suffix to side because both carbons that were aldehydes or ketones are bound to each other. So there's no free former carbonyl groups. So now it's side, to indicate both are non-reducing. That's probably the hardest part. That's usually, people have everything right except the last part. But these others are O's because one of the former aldehydes or ketone groups is free. the reactive pocket. So it's reducing. Okay, so yeah, and I believe in your Dropbox assignment, although I'll remind you after the exam, it's not due for a while. I do have you draw two monosaccharides, but in the stacked conformation, that's the new thing, so you don't have to even flip it. So the trick, you just draw them right underneath each other and you connect them. Sometimes the line is a little bit long and ugly because it depends on if it's a beta. No, because alpha attached to beta then looks really nice, but sometimes it's not. And then it's a longer linkage. Okay, and then I have practice in here to practice naming them. There's also practice drawing, but again, the only one you're going to be drawing is the one in the Dropbox. So that's up to you, but I do have the practice naming in here, and then I do have the answers here. And then the ones that have two names, that means you can name it backwards or forwards. Are there any questions before we cover more complicated sugars? But you're only naming disaccharides. Okay. I have questions after this. Yes. Oh, no, that's good. That's good because if you don't, that's a big stumbling block. Otherwise, in naming it. So if we go back, let's go back. So we go to like, we'll ignore sucrose because that one's split. But if we look at these two, the way we find non-reducing is we find carbon number 1 on an aldehyde. And if that's linked to another carbon of another monosaccharide, it's non-reducing. If carbon number 1 is not free, meaning it's bound to another monosaccharide, that's the non-reducing, and we name it first. If carbon number 1 is free, like in this one, for an aldehyde, then that's the reducing end because it can react with copper because it's not bound to anything. Now for ketones, you look for carbon number 2 and see if it's bound to another monosaccharide, carbon 2, then it's non-reducing. If carbon 2 is free, not bound to anything, like carbon 2, it could be bound to something like carbon 4, but if carbon 2 is not bound to anything, then it's reducing. Yeah. Because it sounds like a little thing, but like as you noticed, if you can't figure that out, then you might name the reverse direction. So here they're showing you microvilli of the intestine and the lumen of the intestine. And so this leads to bigger sugars, which would be polysaccharides. I have a problem with this slide, the use of the word starch. Because they say glycogen and starch, but glycogen is an animal starch. But usually people, when they say starch, they talk about plant starch. The glycogen is animal starch, which is long polymer glucose. When they say starch there, they're talking about plant, but then they really shouldn't do that because there's two plant starches. There's amylose and amylopectin. So it makes it very confusing. So I'm always annoying when someone says, I'm going to buy starch out there. Is that the plant starch? And they don't usually know. So, but all these are starches, okay? They're polysaccharides, so they're long chains. They just all happen to be made up of glucose. So they're all homopolymers, just glucose, 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 glucose. And they all have alpha-1,4 glycosidic bonds. That means carbon number 1 of one monosaccharide is linked to the next monosaccharide with carbon number 4. So it's very repetitive. And they're all alpha linkages, which goes back to, I guess I didn't have to go backwards. But they would look like, they don't have it, data. But we'll see it forward, we'll go forward. all look like this. Okay, now this looks kind of rigid here. Now you'll see some other slides later where they don't have those corners there. And when I was a student, I learned this way, but then when I became a teaching assistant, they now, you'll see other books where they'll smooth it, so it won't look like there's two corners meeting, because you've learned when those meet that usually that means a carbon, but not in sugars. But then later on, like after graduate, they smooth those so it doesn't look like that. Just because people are thinking of carbons. But this is like a building block of some of our starches that are have not just alpha-1,4 linkages, but alpha-1,6 linkages. So, but this down here is shared by all of them: amylose, amylopectin, and glycogen, which I'll be going over. But only glycogen and amylopectin have the alpha-1,6. The reason I say alpha-1,4 is because carbon-1 is linked to carbon-4. Alpha-1,6 carbon-1 is linked to carbon-6. This one would be the non-reducing end. If this is not attached to anything, but the squiggly says it is, but if it wasn't, it would be the reducing side. And again, there's three forms of starch, including the animal starch. But so plant starch, which is glucose polymers, can either be amylose, which is just alpha-1,4 linkages, just a straight line of them. Amylopectin has the alpha-1,6 and alpha-1,4 linkages. Glycogen is like amylopectin, except it branches more often than amylopectin. And that's because the more branches you have, the more non-reducing ends you have. The more non-reducing ends you have, and it makes it easier to break it down for use. And why do animals have more? Because they're the ones usually that are moving. Plants don't usually move. Tumbleweeds don't count because they're not actively moving. They're rolling with the wind or the weather. So amylose is just chain of glucoses, alpha-1,4, no branches. Amylopectin or glycogen, so I only show you one branch, so there's not enough branches to tell them apart. But if you have them side by side, the one with more branches is glycogen. Animal starch. Amylopectin don't look different unless you look at the branches. And they make up your favorite food, maybe not. Well, if you like carbs. But anyway, so pasta, tacos, burritos, anything, bread, everything I like, unfortunately. So this can compare cellulose, well, it's eventually going to compare cellulose to the starch, but not on this figure. They're talking about fiber here, and it can get oxidized to carboxylic acids, and that helps digestion. Just a little health fact. Yeah. And then this is the difference between cellulose, which builds up cell walls in plants and algae, and starch. See, they didn't have again, starch. But that's amylopectin and glycogen to be clear. The glycogen is a form of starch. But notice here, and we'll see other pictures, that if it's not building cell walls and it's used for food, it actually will twist on itself like a helix. Whereas cellulose kind of reminds you of those brick or cement block walls that are built on top of each other. And no one can eat that. Even termites can't eat that. They only can eat that because there's a little protozoan in there helping it eat it. Okay. I don't know if any of you took biology 211 here before the pandemic, where they used to dissect the termite to split and to see the little protist in there that's responsible for breaking that cell. So if they did not have that symbiotic relationship, we wouldn't despise termites as much. And this is, again, this would be starch or amylopectin or glycogen, how it curls on itself like a helix. That's the edible version. And even herbivores really can't eat it without help. Cows have two stomachs. Rabbits have two types of droppings. One they eat, and then they get help from somebody else. Any questions so far? You've been good about asking questions. Some of you have questions later. And we do have one more quiz this week before your exam. It will be due after the exam. And then I don't go into this a lot in great detail, but we did talk a bit in the lipid and membrane chapter. Some proteins have sugar groups on them. And those proteins that have sugar groups on them in a membrane are usually the proteins that are facing the exterior of the cell, so that white blood cells, along with their antibodies attached, can recognize, oh yeah, that's the human membrane versus where the bacterial cell is floating by. We have different distribution of our sugars on our surface. You know, unfortunately, unless you get an autoimmune disease where it can't tell, then that's good. And then proteoglycans means you have some proteins attached to your sugars, and the mucoproteins have a little bit of protein with their sugar as well. But we don't really go into all that. And then just to show you how proteins can be linked to sugars, that's on the next page, but we're not doing any drawings or examining any drawings with that. So O-linked sugars are probably easier to understand because we saw that when you brought two monosaccharides together, they were linked by water and oxygens were involved. So that's probably a little bit easier to understand because here's serine, which you probably remember has a hydroxy group at the end. And when it reacts with this sugar, water was lost. So it seems more logical to have O-linked sugars, where a hydroxy group is with another hydroxy group produce water. But there are some cases where asparagine, and I don't know why it's not glutamine, maybe it's too big, but it's the same structure, where you have an amide linkage reacts with the hydroxy group instead, which seems like not as likely. But these can be parts of what they call a sugar tree, where proteins are covered with these sugars and they branch out and look like a tree. And that, depending on what sugars are there and if they're phosphorylated or not, will dictate where that protein will go to do its function, what organelle it will go to. Like if you take cell biology, you might learn that lysosomal proteins will be phosphorylated on mannose. But that's so that actually those sugars on proteins will also detect where that protein goes in the cell to do its job. Okay, and then this is a picture of a sugar tree. Okay, shown here, where it can be asparagine, where it's end-linked, or where they're both asparagine. But they can be different sizes. But again, if the mannose have a phosphate group on it, it's usually a lysosomal protein. Okay, let's slide it down. Okay, so then, and then some hormones can have sugars attached to them, erythropoietin. Has everyone heard of that hormone? The kidneys make that. So if you have kidney failure, they don't make this hormone, and then your red blood cells count goes down. And then it's a glycoprotein. Okay, so this is showing you erythropoietin, and it has many sugar trees. And then, of course, these sugars, because they have hydroxyl group, would make this protein. Do you think it's soluble in the membrane or in water? Sugars are attached. And sugars are polyalcohols. You think they're going to hang out with fatty acids or water? Water. They're gonna, so, and which is great because we want this protein to travel in the bloodstream, which has a lot of water, all the way from the kidneys to the bone marrow or spleen to make your red blood cells. Okay. And you know, we just went over that just now, asparagine versus serine. Okay. I don't really test you on that. Okay. All right. Oh, and then the slideshow. Oh wow, we finished the whole thing. Okay, so we finished carbohydrates in a day instead of two. Too quiet. Okay. So Wednesday, I think, because I don't want to get too far ahead and talk about something else that's not on your exam. So Wednesday can be optional office hours instead of doing class. So meaning that you don't have to come, or you can come and ask questions. So I don't really want to start glycolysis like 2 lectures early.
Will the Swiss school start though at 6:45?
I think they usually open at 6:20.
Or it's just okay.
Yeah, so it'll still be open then. So it's up to you, so that if you have questions before the exam on Wednesday, you can ask.
Thank you.
You're welcome. I don't really want to start glycolysis super early. So we'll start that the Wednesday after.
You're talking about this Wednesday, right?
This Wednesday would be office hours, and then Monday would be the exam. And everyone knows it'll be the same as last time online. And then a week from Wednesday, I will start glycolysis.
For those of us in BMAC, is the exam already elongated?
I'm sorry.
For those of us in BMAC, do you have the elongated time on the exam?
I should. But I think I spent some time doing that because there's 12 people, so I did start doing that already. But if you see there's a, you should probably, if you log in, it should show you. Yeah, you can check. It should be okay, but it's always good to double check. I'll stop the recording just so we don't record stuff we don't need.